can record. Now it's recording. Okay, now I stop recording. It's on record, and I give the word to Pavel. Pavel, who is, by the way, the working group 4 leader and also the organizer of this webinar series. So please, Pavel, can you introduce? And while we have this introduction for the speakers, I would like to ask the participant to switch off their camera and microphone. Pavel.
Okay, hello everybody. Thank you, Denis. Yes, today's meeting is one of a series of webinars. We have planned a total of 10 webinars. We started in January this year, and the next webinars were scheduled in the following months until the end of this year. Each webinar lasts about 30 minutes with approximately 20 to 25 minutes for a presentation and 5 to 10 minutes for Q&A session and discussion. And the title of today's presentation is Biologics, Industrial Production and of Biopharmaceuticals. And I would like to introduce our speakers for today, Pierre-Mehdi Hammoudi and Gavin Yirajen Vuddamalay from MEB Designing France. Gavin is currently holding his position of head of scientific affairs and business intelligence at MEB Designing, and he's responsible for strategic development and implementation of scientific initiatives, including MEB Design's Watch editions. Gavin holds his PhD in immunology and infectious diseases from the University of Toulouse. Pierre-Mehdi is also part of MEB design team as scientific events manager. He supervises the organization of impactful congress focused on bioprocessing. He obtained his PhD in biology in 2017 at the University of Geneva, and he specialized in molecular biology and infectious diseases. He carried on his academic career as a postdoctoral fellow at the MRC Laboratory of Molecular Biology at the University of Cambridge in the UK. Okay, so Gavin and Pierre-Mehdi, you can start your presentation. The floor is yours.
Yes, so apparently you need to actually stop the sharing so that I can share from my end. Yes, great. Let me do that. This is this. Yes, I'm going to put that on full screen. Yes, so normally you're seeing everything. Yeah, the cursor is here, so everything. Let me know if there's an issue with the slides. Thank you for this kind introduction. Pierre-Mehdi joins me to thank you on behalf of Map Design to allow us to actually host this webinar on the industrial production of biopharmaceutical. As an introduction, a few words about Map Design. So we are a French organization. We've been around for 10 years now. And our sole purpose is to support and promote the biopharmaceutical and bioprocessing sector. So we do that through various actions and services. And we've been working with both the industry and academia. And for a few years now, we have a growing number of institutions. So talking about biopharmaceuticals, we can subdivide that into three main categories. So first, the therapeutic proteins, where we find the antibodies in various formats and specificity of the proteins like enzymes, cytokines, and hormone. Then we have the vaccines, the therapeutic ones, which are primarily used in oncology, and the prophylactic one used exclusively, in infectious diseases. Then we have the last group, which are the ATMPs for advanced therapy medicinal products, where you will find the cell therapies, various types of gene therapies, and finally, tissue engineering. So to date, biopharmaceutical represent only 4% of the global arsenal of drug products that are available to cure or prevent diseases. The 96 remaining percent is being taken up what we call by small chemical molecules. And things get interesting when now we look at the pipeline, which correspond to both the clinical and non-clinical phases of drug development. And taken together, biologics are at 61% and small molecule at 39%. So we have more biological products being developed as compared to small molecules. And this has been the case for a few years now. And just below, you have an idea about the proportions between therapeutic proteins, ATMPs, and vaccines. So now if I have a look at really the basis of these biological products, of course, if you are using cell therapy, you're going to have cells that are injected to the patient. If you're using gene-modified cell therapy, also known as ex vivo gene therapy, like CAR-Ts, you're going to use cells again, which are now modified. And beyond these cells, we can also use proteins, organelles, or vesicles that are derived from these cells. And through genetic engineering, we can actually modify these cells so that we can actually make them produce therapeutic products like antibodies, viral vectors in the form of viruses, and various types of nucleic acid. So basically, the cell and the subcellular components of the cells are considered as biopharmaceutical. So now, if we compare these biopharmaceutical to their small molecule counterpart, you can see that when we move from nucleic acid to stem cells, cell-based therapies, etc., we are increasing the degree of complexity of the drug product. And this increase in complexity and molecular mass will, of course, influence the way we produce these drug products. So when we talk about the industrial production of biopharmaceutical, also known as biologics, we use either the term bioproduction or more commonly the term bioprocessing. So we're going to see that bioprocessing involves the use of several biotechnological tools and also genetic engineering. And basically, we can actually separate bioprocessing steps into three main steps: the upstream process, the downstream process, and finally, the fill and finish process. So as you might have guessed, the cell has a central position in this bioprocessing steps. So of course, if you are producing cell therapies, we're going to expand cells, but also if you are producing subcellular components like DNA, nucleic acid, proteins, etc., we're also using the cells as what we call an expression system. Okay. So if now you've been involved in cell culture, you know that we need to provide the cells with enough nutrients for them to grow. And with these nutrients, we need also to provide what we call culture parameters. In the bioprocessing field, we call that critical parameters in terms of temperature, CO2 levels, things like that. And as the cells will grow, of course, the amount of therapeutic products will increase in this cell culture. But in parallel to that, you also have waste in terms of cellular debris, toxins, etc. And if you are looking at an industrial process, these waste need to be eliminated so that at the end we get a pure concentrated sample of the drug product. So talking about expression system, there's a lot of different cell types that can be used. Most of them are cell lines. You can use mammalian cell lines to produce antibodies, in cell lines to actually produce viral vectors in the form of AAV, bacteria to produce, for example, insulin. And we also have examples where we use algae as a source for biologics. Here, I would like to point out that we have now emerging technologies that do not use cell at all. They are known as cell-free technology, where we don't use a cell, but we use components of the cell to produce the nucleic acid or to produce the therapeutic protein. So what we're going to see, this is a generic diagram of the bioprocessing stages. The raw material most of the time would be cells, so either cells will be engineered or not. And then we're going to grow these cells in what we call bioreactors. This will be the upstream process. There will be an intermediate step of clarification, which is known as drug recovery. And then we're going to move to the downstream process where actually we'll be removing any impurities, aggregates, etc., to finally sterilize the drug product and condition it, formulate it in a way to be administered to the patient. So the upstream process, up means increase in volume. So basically what we're going to do is we're going to start with a small sample of cells, so it can be a cell line or primary cell, and through various sequences, we're going to increase the volume of the culture. And when we increase the volume of the culture, of course, we're going to increase the number of cells, number of product that these cells might be producing. But in parallel, there will be also a buildup in biological waste and toxin. And so these steps where we're going to increase gradually the volume can also known as scale up. And this is a very essential step because if you also remain involved in cell culture, and if you put 1 ml of cell culture in 1,000 liter of medium, all the cells will die. So if you really want to go from millimeters to hundreds or thousands of liters, you will need to go through this upstream process of scale up and increasing the volume. So once we have enough cell or enough product in the medium, we move to the downstream process. So basically here we're going to do, we're going to recover, concentrate, and purify the drug product, whether it's a cell or it's a subcellular component. And there are a lot of steps that are involved. Some are specific for the biologics that you want to produce. But basically, you could actually separate that into the harvest, where the drug recovery, the purification, where you would eliminate the contaminants, and the polishing, where you would, for example, eliminate remaining impurities or aggregates, especially if you're working with proteins, a lot of aggregates during the purification process. So, and as you can see here, the aim of the downstream is to go down in the volume. So when you go down in the volume, we are increasing the concentration of the drug substance and we're also purifying it. And the final step would be the fill and finish step. So you have your sterile drug substance, you're going to formulate that, fill it in an aseptic way. You can, depending on the biologics, do a lyophilization and finally package that to be actually sent to the patient. And obviously here, if you are working, for example, with enzymes, you can go through lyophilization. Antibodies can be actually conditioned in vials, but if you're using, for example, cell-based therapies, you will use IV bags to be administered to the patient. I have taken a few specific examples of biological products, starting with monoclonal antibodies. So here, basically for antibody production, we will start with a cell line. So this cell line will be modified to express a transgene that would code specifically for the antibody. And here, the gold standard in antibody production would be CHO cell lines. So once you have these modified cell lines, you're going to start your culture. And throughout the upstream process, there are several options, choices to be made. So first is whether you're looking at single-use cell culture or cell culture in stainless steel. So for single-use, everything will happen in bags where the culture will take place, and these bags have connecting tubes for the technicians to feed in nutrients or remove liquids, etc. And then you can choose also not to use single-use bags, but use directly the stainless steel bioreactor, and everything goes on into this bioreactor. And here, the difference is that the carbon footprint is not the same if you use single-use or if you use stainless steel. So once you made that decision, you move now to the culture itself. Again, several technologies are now available where you can use batch culture where the volume is constant. You can now use feed batch culture where you will add additional fresh medium throughout the cell culture. Or you can move to continuous culture where there will be a continuous harvest of old medium and addition of new medium, but the volume will be constant. So once you reach the amount of antibodies that you want, you now move to the downstream step, where here you will use like various chromatographic columns, protein A columns to capture your antibody, and then you move on to the rest of the downstream process, remove any eventual viruses that have been present in the cell culture, and then you have your product. Here, you need to take into account that if you are working with conjugates, you will have additional step after this one, where, for example, you need to conjugate your antibody with radioligands for radiopharmaceutical or drug molecule for ADC. If we now take the example of viral vectors, same thing. In the upstream process, we are going to use cell lines. These cell lines are known as packaging cells, where we're going to use various combinations of plasmid to transfect these cell lines, and then the cell will start producing a virus, which will correspond to viral vectors. And again, through various scale-up of the cell culture, you will end up with a sufficient amount of virus, and then you will move to now the DSP process, where you will purify these viral vectors. And you can see the difference between antibody production and viral vectors. One, you have to remove the virus, and here you need to actually harvest the virus and purify this virus. Concerning cell therapy, once again, we will start with cells. These cells can be fresh or frozen. There's always a preparation, purification step to have like a concentrated amount of sample. And then through various activation process using antibodies, cytokines, etc., you're going to expand these cells. This corresponds to the upstream process. Again, once you have reached the sufficient amount of cells, you will go through the downstream process to purify your cells, remove any dead cells, debris, etc., and then formulate that either in vials or IV bags to be administered to the patient. Finally, if you are producing gen-modified cell therapies like CAR-Ts, you will have to add an additional step where you need to modify the raw material. So this can be done before the upstream process, or it can be done also during the upstream process, where you're going to use either viral vectors or non-viral vectors to actually modify these cells. And then we move to the rest of the classic upstream process, expansion, downstream with purification, and finally fill and finish. So when we are talking about cell-based therapies, what is important is to consider the source of the cells. So you can either have autologous source, where here you're starting with the patient, take a sample of cells from this patient, and then you modify, expand them ex vivo, and then you take back these cells to the patient. So of course, this is a patient who is sick, and time is of the essence, and you cannot have extended time for bioprocessing. So most of the time, for example, for CAR-Ts, we're looking at something which is under 3 months of bioprocessing steps. And then another source would be allogenic. So here we're not starting with a patient, but we're starting with healthy donors. And again, once you have a sample of cells from healthy donors, you can expand them, modify them, etc. But here you can also store them. So you can freeze them and have what we call off-the-shelf products, where once you need patients, and here you don't have to treat only one patient, you can treat several patients. Once you need to treat patients, you can send these products to these patients. So I would now leave the floor to Pierre-Mehdi.
Thank you, Gavin. So now that we have reviewed how different classes of biologics are produced, we'll move to how to go through industrial scale production, to true industrial scale production. And to do that, we'll cover a couple of aspects. So first, some key regulatory and technical concepts such as CMC and GMP. Then the current trends that are shaping the biomanufacturing industry today. And finally, a closer look at the French ecosystem and its capabilities. So first, CMC. So the concept of CMC, which stands for chemistry, manufacturing, and control, is central to the development of any type of biologics. So CMC defines the product, the process, and the testing strategy that together will ensure that every batch is safe, effective, and consistent. And so unlike
small molecules, biologics being living products, there's a need for CMC documentation to describe the expression system, the purification strategies, the analytical controls, and the product, any product specification in great detail to achieve a robust and sustainable production. So CMC basically is the scientific foundation that ensures reproducibility and safety before entering into GMP production. So then comes the GMP for good manufacturing practices. So while CMC defines what must be achieved, GMP defines how we must operate to achieve it. And so basically, GMP focuses on different pillars, such as product quality, basically to avoid contamination and ensure consistency in the identity and the potency of the batches. Patient safety by validating every step from sterilization to equipment calibration, for instance, and also regulatory compliance, since the GMP certification is mandatory to obtain market authorization. So you can think of GMP as the execution layer that translates CMC principle into daily operations on the facility. And it's important to note that both CMC and GMP overlap, but they are not identical. So CMC is product specific. It demonstrates the process understanding and control, while GMP instead is facility oriented, and it ensures that the quality of the system functions properly. So take an example, a manufacturer may have an excellent GMP system overall, but each new biologic will require its own CMC package. And so bridging these two levels, scientific and operational, CMC and GMP, is actually one of the most demanding aspects of biologics manufacturing. And so on the next slide, you can see that some of these principles are framed by international regulatory guidance. On this slide, you have example of the FDA and the EMA. That basically this kind of guidance covers everything from characterization of the active substance to comparability studies or validation of process changes. And the key message is that the regulatory expectations are harmonized globally, but biologics, because of their nature, still demand a case-by-case approach, because no two cell lines are exactly similar, no vectors behave exactly the same way. And so these challenges there are a number of challenges that are raising because of the emerging therapies, especially I'm thinking here of cell and gene, for instance, that are constantly pushing the regulatory standards to stay up to date. And so in this respect, there's a need for the regulatory agencies to include experts in their panels. There could be people in the audience here that have already acted as such experts, or that could be susceptible to do it in a close future, to basically question and tackle appropriately the regulation of existing biotherapies and those in development. And so the last part I would like to emphasize here is that beyond manufacturing, quality control and analytics represent a major share of both cost and risk in the development process. So if we take the example of monoclonal antibodies, for instance, QC alone may account for about 30 to 50% of the cost of a production run, so that's quite substantial. So there's a need for every company to develop its own analytical strategy to complement the regulatory standards and define precisely which attributes are critical for quality, but also purity and potency of each biologic and in each batch. And as shown on this example for antibodies, there are multiple layers of assays that are needed to characterize structurally, functionally, also pharmacologically the biologics. And so these depths of analytical tools is what will allow the manufacturers to truly understand their products' behavior and ensure consistency over the long term. So to the next slide. So we have seen in the previous part some of the core features of biomanufacturing, and it's important maybe to make a stop on the current trends in the industry. We could have mentioned more points here, but we picked two. First one is sustainability. As it was mentioned by Gavin, this is a key feature that to ensure that the supply chain is resilient and responsible in terms of impact on the environment. There's also a concept of cost here that enters into account. And so there are a number of initiatives that have been carried out by manufacturers, such as localizing the production, decarbonizing the transport using lesser or greener packaging, but also overall optimizing the production and manufacturing methods to achieve an answer to a goal actually that the societies are pushing for lesser footprint of the production of biologics. So the second point is the digitalization and automation. So technologies such as digital twins, AI, of course, AI-driven process optimization, robotic facilities are redefining over the last years, and they have contributed to redefine how we monitor and control bioprocesses. And so the purpose of these tools is to help to reduce variability, predict more accurately deviation, accelerate tech transfer between sites, which is a very impactful, could be very impactful for a manufacturer. And so the purpose here is simply to raise your awareness on the fact that we've observed through our different activities that these are topics that have received increasing attention over the last years, and it's probably only the beginning. So we last wanted to give you a snapshot of the French ecosystems in terms of production capacity. So France, over the last year and decade, has built a relatively dense and diverse biomanufacturing ecosystem, and that's an example in Europe in many sides. So on this slide, you have a picture of the CMO and CDMOs capabilities and distribution across the country around regional hubs near to Paris and Lyon, of course, but also in area in southwest of France. And so many of these sites operate at clinical and commercial scale already, offering an expertise in various types of biologics, such as monoclonal antibodies, of course, but also viral vectors and TMPs more broadly. And so we wanted to complement this picture with another layer of the ecosystem that we believe is quite important and that has emerged over the last 5 years or so, which are the so-called industrial integrators, which are academic platform mostly, that were labelized, certified by the French government in order to make the link between academic research and industrial partners. So in order to help to prepare and de-risk projects emerging from academia and get them ready as possible for the future steps that we'll encounter into during their industrialization. So together, these two slides illustrate how France is positioning itself as a key European player in the biologics manufacturer with its capacities that represent to date there are at least 20 CDMOs that have a production capacity in France with over 30 sites. So nearly half of these sites operate already at the commercial scale, and new projects, especially I'm thinking of cell and gene therapy projects, are under active discussion and will continue to impact and reshape the ecosystem. So the purpose here is to highlight that the landscape is expanding rapidly depending on the needs of the ecosystems that are emerging underneath. There's increasing need of adaptability of the facilities to respond efficiently to, I'm not talking even about pandemics, but at least to the evolving needs of the industry. So a few take-home message to conclude this section and overall the presentation. So we have seen that therapeutic proteins remain to date the most mature and cost-efficient technology. It's also true that cell and gene therapy, despite the fact that they are still at an earlier stage in terms of industrial development, they are so scientifically promising that they will necessarily gain there will be more and more projects to using cell and gene therapies, but still today high production cost and the complexity to scale up the production is an impactful challenge. And so there is probably for the next decade the key challenge is to industrialize these new modalities efficiently in order to make them as reproducible, affordable, and also sustainable as perhaps the antibody platforms that we know today. And perhaps one last word about for those of you who would like to explore further this concept and these topics. MapDesign is offering a free resource platform which is called the BioHub, which is available on our website, and it includes monthly pipeline updates, KOLs interviews, watch reports, bioprocess, and immunowatch. The next one is about to come out in a few days. So new content is announced on our different channels, LinkedIn, website, and newsletter, for instance. So do not hesitate to follow us to stay connected with the latest developments in biomanufacturing, but not only. It's worth to mention, and that's probably one of the bridges with Enota, that among the events that we organize, we have one focused on antibodies, the Antibody Industrial Symposium, to which Enota has been a contributor over the last year. And the next event is going to take place at the end of June, beginning of July 2026 in south of France, in Montpellier. So we'd be pleased to welcome you, of course, but already to let you know about the content of this event if you have any questions. So that brings me to the end of this presentation. So I'd like to thank you all for your attention, and we'll be very happy with Gavin to take any of your questions.
Okay, thank you very much for your presentation. And now we have a Q&A session. So please, the audience, ask any questions and just thoughts in relation to the presentation.
And you can open your camera if you want, and your microphone. Just to start, you mentioned sustainability of manufacturing these therapeutic antibodies production. How much do we know if we compare biopharmaceuticals and small molecule drug in terms of manufacturing impact on, let's say, carbon footprint, for instance?
So that's a very good question, and I think a lot of the answer relies. I mean, for small molecules, the processes are probably established since a longer time. So I guess the footprint here is directly depending on the transportation, the packaging, and so on, rather than on the production itself and the development, at least itself. While it's probably more the case still for many biologics, because there is probably a larger need in terms of media, in terms of so I would say that probably to date there is still a high cost in terms of development associated to the footprint of biologics, and still as Gavin mentioned, some questions about the single use, the different kind of batch production. This will have necessarily an impact, and the idea with the new tools can bring here is to de-risk as much as possible and to evaluate as early as possible what could be the alternative to a given process established in a certain way.
You also need to take into account the additional steps because when you compare chemical synthesis to actually the bioprocessing steps, you have additional steps, and sometimes it's also the additional steps that need to be taken into account, and this actually increase the carbon footprint. And basically now you have companies, consulting firms that actually are experts in assessing the carbon footprint of each type of biopharmaceutical. And here it's important to take into account also the volume. So when you're looking at, for example, CAR-Ts, which are autologous cell therapies, we are producing in liters of medium, while looking at antibodies, we're looking at 10 to thousands of liters of medium. So there's a large spectrum of carbon footprint based on the biologics that you are producing itself, but definitely it's well higher than simple chemical synthesis, especially because, well, the chemical synthesis is now quite mastered, so we know where to actually cut the carbon footprint.
Thank you, Gavin. Thank you. Any other question in the audience?
I have one very one question, and please answer shortly because we are pretty lag of time. The question is, what about cost of biologics production? Do they decrease in coming years to make the production of biologics more common? How do you think?
So this is a very important question because we have the cost, which is the selling price, so what the biopharma companies sell their product, and then we have the cost of goods, which is known as the COG. So the COG is something which is highly confidential. This is something that Map Design has been trying to have access for years. We know most of the CDMOs in France are members of Map Design, but this is something they will not disclose. So there's a big difference between how much money it's costing to produce that and how much money they are actually selling it. Then comes how mature is the technology. So, for example, antibodies, the gold standard is CHO cell lines. We're looking at between 10 to 15 grams of antibodies produced by liters. And well, this is where actually the fact that it is more mature, this is where we are reaching the cost. Then we need to take into account also the biosimilars. So there are biosimilars for antibodies. You can reduce the cost by at least one log, so 10 times less when you are producing biosimilars as compared to actually the novel ones. If you compare what for example, Asian countries like India are producing, we are already beyond one log of difference between the price biosimilars and the novel ones. Then come the question of cell and gene. So cell and gene will range from maybe can go up to 4 million euros for the single dose. But here the price is not indicative of the cost of good. The price is indicative of the therapeutic value. This is where actually the whole question of pricing comes in. If you have one injection and you cure someone that would have a chronic disease for the rest of its life, of course, the therapeutic advantage is there, and so they can have this price. But basically, antibodies, most probably, we're looking at a gradual decrease in the prices. But for cell and gene, it's something which is completely different.
Okay. Okay, thank you very much for the answer and for the answers, I guess, because we have to
We
finish.
have one last question. I think Albino raised his hand. So Albino, you want to ask a question?
Yeah, if it is some time remaining, it's a fast question, in fact, because in the tissue engineering products, there is sometimes not just cells with the materials, but in fact there are some others biologicals involved in this type of products. How it is the framework of this type of products, combining cells and biologicals in the same final product, and by materials also, in fact.
Sorry, I didn't understand the question. So I understand that you're talking about combination of biopharmaceutical, and the question was about
how it is in regulatory terms, how it is
okay.
the
So
framework with biomaterials, biologicals, and cells.
yes, so
It's a
here
combined product,
it's
right?
under yeah. The answer is that you have a specific regulation when you have combinations of biologics. So this is taken into account under the ATMP regulation. So ATMP regulation says that any combination of cell and gene therapy has a specific guideline. So you would find that the combination for that. And I know that in France you have a special agency known as the IAS that helps you actually define what type of product in terms of regulatory aspect. And this is a very important question because for the novel therapies, sometimes people don't understand, are we working with cell therapy? Are we working with gel therapy? Is it a combination of that? And this will greatly influence how the CMC and the GMP and any regulatory findings. And well, in France, we know that people help us for the regulatory aspects, but then you can have a scientific advisor from EMA, FDA have that. They have a specific actually booth for you to contact them and maybe define that. So regularly you will find Excel sheets provided by the EMA, and they will say, okay, if you're working, for example, at IPCS and you combine that with tissue engineering, this is the regulatory category that you want to use.
Okay. Thank you,
So,
everybody, for for the answers and for the brilliant presentation to both of you. And okay, the next and the last one webinar is scheduled on 18th of November 2025 from 5 to 5:30 p.m. set, and our speaker will be Sean Gavin from the University of Manchester from United Kingdom. And the title of this webinar will be Markov Models, Simple Practical Examples. So I would like to invite you to participate the event. Please register for the webinar. And thank you for attending and participating this webinar. Thank you. Bye.
thank you all.
Thank you.
Goodbye.
Bye bye.
Bye bye.